Get ready for a deep dive into the world of CRISPR.
Yeah.
We're going to be exploring this revolutionary gene editing technology, its potential to cure diseases, and the stories behind it.
Okay.
Your source material for this one, excerpts from Walter Isaacson's The Code Breaker.
Oh, yeah.
It's all about Jennifer Doudna,
Right.
a leading figure in CRISPR.
Isaacson's book is really good. It weaves the science of CRISPR with the human side of discovery.
Right.
You get a real feel for the drive, the setbacks, and the collaborations that lead to breakthroughs.
It's not just a science textbook.
Right.
It's a story.
Yeah.
And speaking of stories, let's start with Doudna's.
Okay.
Isaacson takes us back to her childhood in Hilo, Hawaii. Apparently, even as a kid,
Yeah.
she was drawn to the mysteries of nature.
There's this anecdote about sleeping grass that fascinated her.
Oh, wow.
How it would fold up when touched. It's almost foreshadowing her work with RNA, this tiny thing controlling a plant's response. It shows that big discoveries can start with the smallest observations.
That's so cool. And her interest in science really took off when she read The Double Helix by James Watson.
Oh, yeah. Great book.
Imagine a book sparking a passion that leads to a Nobel Prize.
That book tells the story of discovering DNA's structure.
Right.
It's a classic. Doudna's journey shows how one discovery builds upon another.
Right.
Pushing the boundaries of what's possible.
Right, like standing on the shoulders of giants. Now, Doudna's PhD work focused on RNA,
Okay.
a molecule closely related to DNA. You could say DNA is the blueprint of life,
Yeah.
and RNA is like the messenger carrying instructions from the blueprint to the rest of the cell.
Exactly. And her mentor, Jack Szostak, believed that RNA held clues to the origins of life itself.
Wow.
This shows how curiosity-driven research, even if it doesn't seem immediately useful, can pave the way for incredible advancements.
It's like exploring a new continent. You never know what treasures you'll find.
Yeah.
And Doudna's personal life was deeply intertwined with her work.
Okay.
Around the time she made a major breakthrough, her father was diagnosed with cancer.
It's a reminder that scientists are real people, driven by the same hopes and fears as everyone else. Sometimes personal experiences can fuel their determination to solve the world's biggest problems.
Now, let's get to the CRISPR breakthrough itself.
Okay.
It all started with a scientist named Francisco Mojica,
Right.
who discovered repeating DNA sequences in bacteria, something nobody had seen before.
Initially, nobody knew what these sequences, later named CRISPR, were for.
Okay.
But Mojica kept digging. He eventually realized that CRISPR was part of the bacteria's immune system,
Oh, wow.
like a memory bank of past viral attacks.
Pretty amazing, huh?
Bacteria with an immune system, who knew?
Yeah.
Doudna was hooked by this discovery.
Okay.
She teamed up with Emmanuel Charpentier, a scientist with a different but complementary expertise.
To unravel the mysteries of CRISPR.
That collaboration was key.
Yeah.
Charpentier was struggling to understand a molecule called trachRNA,
Right.
which turned out to be crucial to the CRISPR system. Together, they figured out how it all worked.
This is where it gets really cool.
Yeah.
One of Doudna's team members,
Okay.
Martin Janek,
Right.
had this aha moment. He suggested simplifying the system by combining two RNA molecules. And that's how they created the powerful gene editing tool we know as CRISPR today.
Their 2012 paper describing this simplified system was a bombshell.
Wow.
It set off a race to make CRISPR work in human cells,
Right?
opening up incredible possibilities for treating diseases.
Imagine being able to edit the very code of life. It was like discovering a new element, a fundamental building block of biology.
And the race was on.
Yeah.
Feng Zhang, a brilliant young scientist, also figured out how to make CRISPR work in human cells. He, along with George Church,
Right.
published their findings almost simultaneously with Doudna's team. It was a scientific photo finish, highlighting the intense competition in cutting-edge research.
That kind of competition can be fierce.
Yeah.
Especially when patents and potential profits are involved.
Right.
There was a major legal battle over who would control the rights to CRISPR technology.
This raises questions about who owns scientific discoveries and how to balance the need for profit with the pursuit of knowledge for the benefit of humanity.
Right.
It's a debate that continues today.
But before we get into the ethics of it all,
Okay.
let's talk about the potential downsides.
Okay.
Gene editing is powerful, but it's not without risks. The story of Jesse Gelsinger is a stark reminder of that.
Gelsinger was a young man who tragically died in a gene therapy trial in the late 90s.
Oh, wow.
This event was a major setback for the field. underscoring the need for extreme caution when manipulating the human genome.
His death forced everyone to take a step back and acknowledge the ethical complexities involved.
Right.
It's a reminder that even with the best intentions,
Yeah.
there can be unforeseen consequences.
It was a wake-up call.
Yeah.
But it also paved the way for more rigorous safety protocols.
Right.
And a deeper understanding of the ethical considerations surrounding gene editing.
And with CRISPR, the ethical stakes are even higher.
Right.
We're not just talking about treating diseases anymore. We're talking about the potential to alter human evolution itself.
That's where the debate about germline editing comes in.
Right.
This is the idea of making changes to DNA that can be passed down to future generations.
Wow.
It raises all sorts of ethical questions
Yeah.
about designer babies and the potential for unintended consequences that could affect generations to come.
It's like stepping into uncharted territory.
Right.
And the story of He Jiankui, the Chinese scientist who created the first gene-edited babies,
Right.
really highlights the urgency of these ethical concerns.
Jiankui's actions were met with widespread condemnation.
Yeah.
He was ultimately imprisoned by the Chinese government. This case showed that the scientific community needs clear guidelines and international oversight to prevent rogue scientists from crossing ethical boundaries.
It's a stark reminder that scientific progress can't happen in a vacuum. We need to consider the potential consequences of our actions,
Right.
not just for ourselves, but for future generations.
Absolutely. But amidst all these ethical dilemmas, it's important to remember the incredible potential of CRISPR to alleviate human suffering.
Right.
The story of Victoria Gray, who was successfully treated for sickle cell disease using CRISPR, is a powerful testament to that.
Here's a woman who is facing a debilitating, life-shortening disease.
Right.
And thanks to CRISPR, she's now able to live a normal life. It's an incredible story of hope and a glimpse into the future of medicine.
Her story is just one example of how CRISPR could revolutionize healthcare. Scientists are exploring ways to use it to fight cancer, treat blindness, and potentially cure a wide range of genetic diseases.
It's mind-blowing to think about what might be possible in the coming years. But this power also brings a responsibility to consider access and equity. We need to make sure these treatments are available to everyone, not just the privileged few.
That's a crucial point.
Yeah.
CRISPR therapies have the potential to be incredibly expensive.
Right.
We need to find ways to make them affordable and accessible to all,
Right.
regardless of their economic background or geographic location. Otherwise, we risk exacerbating existing health disparities.
It's a complex issue, but one that we absolutely need to address. And speaking of pushing boundaries, there's a whole movement of biohackers out there.
Right.
People like Josiah Zayner, who believe in democratizing gene editing and taking it out of the hands of big institutions.
Zayner represents a DIY approach to gene editing. He's even experimented on himself. It's a fascinating and somewhat unsettling trend. It raises questions about who should have access to this powerful technology and what level of regulation is appropriate.
Even Jennifer Doudna herself, who was initially very cautious about gene editing, has evolved in her thinking. She's become a leading voice in the ethical debate, acknowledging the potential benefits while advocating for responsible use.
Doudna's journey reflects the evolving understanding of CRISPR's power and the responsibility that comes with it. She recognizes that we can't just focus on the science. We need to have open and honest conversations about the ethical implications and establish clear guidelines for its development and use.
And speaking of a world in desperate need of solutions, the COVID-19 pandemic really brought CRISPR research into the spotlight. It highlighted the need for new approaches to fighting infectious diseases. And CRISPR provided some promising answers.
The pandemic was a catalyst for innovation, particularly in the development of rapid diagnostic tests. CRISPR-based tests like Detector and Sherlock were game changers in the fight against COVID-19. They provided fast and accurate results, which was crucial for controlling the spread of the virus.
It's incredible how quickly these tests were developed. And the pandemic also spurred research into new types of vaccines, including the revolutionary RNA vaccines. which were highly effective against COVID-19.
RNA vaccines are another example of how scientific breakthroughs can happen rapidly when there's a global crisis. They've opened up new possibilities for vaccine development,
Yeah.
not just for coronaviruses, but for other infectious diseases as well.
It's amazing to think that CRISPR itself could be used to directly target and destroy viruses.
Right.
Imagine a future where we can use this technology to fight off infections before they even have a chance to take hold.
That's the idea behind CRISPR-based antiviral therapies like Carver and Pac-Man.
Okay.
They're designed to target and destroy the virus's genetic material,
Right.
essentially disabling it. It's a whole new approach to treating viral infections, and it has the potential to be incredibly effective.
It's like having a molecular scalpel that can precisely excise the viral code from our cells. The possibilities are mind-boggling, and the pandemic really showcased the collaborative spirit of the scientific community.
Yeah.
Scientists from all over the world were sharing data, working together,
Right.
and pushing forward at an unprecedented pace.
It was a truly inspiring example of global cooperation in the face of a common threat.
Yeah.
Scientists set aside their individual ambitions and came together to solve a global crisis. It was a reminder that science can be a unifying force for good. It was a remarkable display of scientific collaboration, and that same spirit of innovation is driving the future of CRISPR. It's not just about treating diseases anymore. We're entering a new era of biology where we have the power to manipulate the very building blocks of life.
It's like we've unlocked a secret code, a language that allows us to rewrite the instructions of life itself. Doudna's Nobel Prize win certainly recognizes that.
Yeah.
And her predictions about a future where biology is more accessible and collaboration is more global are both exciting and a bit daunting.
It's exciting to think about the possibilities.
Right.
But we need to be mindful of the potential risks as well.
Right.
CRISPR is a powerful tool.
Yeah.
It needs careful consideration and ethical development to ensure its benefits are shared equitably and don't lead to unintended consequences.
Okay, so we've talked about the history, the ethical dilemmas, the potential applications.
Right.
But let's dive a little deeper into the actual science of how CRISPR works.
Okay.
It's pretty amazing stuff.
It really is. At its core, CRISPR is a system that bacteria use to defend themselves against viruses. Think of it like a bacterial immune system. Remember those repeating DNA sequences Mojica discovered?
Yeah.
They act like a genetic library of viral DNA fragments.
So the bacteria keep a record of the viruses they've encountered. It's like a molecular mugshot book.
Exactly. And when a virus attacks again, the bacteria use those stored DNA fragments to create short RNA molecules. These RNA molecules act as guides, leading a special enzyme called Cas9 to the matching viral DNA.
So it's like a wanted poster for the virus, and Cas9 is the bounty hunter.
That's a great analogy. And Cas9 is like a pair of molecular scissors. Once it's guided to the viral DNA, it makes a precise cut, disabling the virus and protecting the bacteria.
That's so cool. It's like a search and destroy mission at the molecular level.
But how did scientists figure out how to use this bacterial system for gene editing in human cells?
That's where Doudna and Charpentier's breakthrough comes in. They realized they could reprogram the CRISPR system by creating synthetic guide RNAs that would target specific DNA sequences in any organism, not just viruses.
So instead of targeting viral DNA, they could target any gene they wanted. Precisely. It was a game changer. They essentially turned a bacterial defense mechanism into a powerful gene editing tool that could be used to target almost any gene in any organism.
That's incredible. So in the case of Victoria Gray, who was treated for sickle cell disease, how did CRISPR work its magic?
Sickle cell disease is caused by a single mutation in a gene that's responsible for making hemoglobin, the protein in red blood cells that carries oxygen.
Right.
Scientists used CRISPR to edit stem cells in Gray's bone marrow, correcting that mutation.
Now, for anyone who's not familiar with stem cells, they're like the master cells of the body.
Right.
They can develop into all sorts of different cell types, including blood cells.
Exactly. So by editing the stem cells, they ensured that Gray's body would continue to produce healthy red blood cells with normal hemoglobin, effectively curing her of the disease.
That's revolutionary. It's like rewriting the script of her life.
Yep.
And that's just one example of the many potential applications of CRISPR. But as we've discussed, this power comes with a huge responsibility, Fader.
Absolutely. We need to be extremely careful about how we use this technology. There are ethical considerations, safety concerns, and questions about access and equity that we need to address.
We've talked about the ethical dilemmas of germline editing and the potential for designer babies.
Right.
But there are also concerns about the potential for CRISPR to be used for enhancement
Right.
rather than just treating diseases.
The idea of using CRISPR to enhance human capabilities like intelligence, strength, or lifespan
Right.
is both fascinating and ethically fraught. It raises concerns about creating a society where genetic advantages are available only to those who can afford them,
Right.
further widening the gap between the haves and have-nots.
It's a slippery slope. But putting aside those concerns for a moment, what are some of the most exciting developments on the horizon in terms of CRISPR research?
Well, as we mentioned, CRISPR is already being used in diagnostics with rapid tests for viruses like COVID-19. But researchers are also exploring ways to use CRISPR to treat a wide range of diseases, from cancer to genetic disorders.
And they're even looking at ways to use CRISPR to modify crops,
Right.
making them more resistant to pests and diseases,
Yeah.
and potentially even increasing their nutritional content.
That's right. CRISPR could revolutionize agriculture, helping us to produce more food with fewer resources,
Right.
which is especially important in a world facing climate change and a growing population.
It's clear that we're just at the beginning of the CRISPR revolution. The next few decades are going to be incredibly exciting
Yeah.
and potentially unsettling
Yeah.
as we grapple with the implications of this powerful technology.
It's a journey into uncharted territory.
Yeah.
CRISPR has the potential to transform medicine, agriculture, and even our understanding of evolution itself.
Right.
It's a powerful tool that could be used for both good and ill, and it's up to us to decide how we want to shape this future.
That's why it's so important for everyone to engage with this topic, to learn about the science,
Right.
to understand the ethical dilemmas,
Yeah.
and to participate in the conversation about how we want to use this incredible technology.
Knowledge is power.
Mm-hmm.
The more we understand about CRISPR,
Yeah.
the better equipped we'll be to make informed decisions about its development and use.
Isaacson's book really emphasized how CRISPR didn't come about because scientists were specifically trying to create a gene editing tool.
Right.
It was a result of curiosity,
Right.
of scientists just trying to figure out what these strange repeating DNA sequences were doing in bacteria.
It's a perfect example of how fundamental research,
Yeah.
driven by curiosity and a thirst for knowledge,
Mm-hmm.
can lead to completely unexpected and revolutionary discoveries. It's a reminder that we should never underestimate the power of asking questions and exploring the unknown.
And Doudna's own story, with her childhood fascination with sleeping grass, is a testament to that.
Yeah.
It shows that scientific curiosity can start at a young age and lead to extraordinary places.
It's a reminder that science is not just about facts and figures.
Yeah.
It's about a sense of wonder,
Mm-hmm.
a desire to understand the world around us,
Right.
and a willingness to keep asking questions even when the answers seem elusive.
It's about embracing the unknown and never losing that childlike sense of awe and excitement about the natural world. And it's a reminder that anyone can be a scientist,
Right.
regardless of their background or formal training.
All it takes is a curious mind and a passion for discovery.
Another theme that stood out in Isaacson's book is the collaborative nature of scientific research.
Mm-hmm.
CRISPR wasn't the product of one brilliant mind working in isolation.
Right.
It was the result of countless scientists building upon each other's work,
Right.
sharing ideas,
Yeah.
and working together to solve complex problems.
The story of CRISPR is a testament to the power of teamwork. Science rarely happens in a vacuum.
Yeah.
It thrives on the exchange of ideas,
Right.
the sharing of data, and the collective effort of researchers from around the world.
And it's not always smooth sailing, right?
Right.
There's competition,
Yeah.
disagreements, even clashes of ego sometimes.
Mm-hmm.
The patent battle between Doudna's team and Zhang's team is a prime example.
It's true. The competitive nature of science can be both a driving force for innovation and a source of conflict. But ultimately, science is a human endeavor,
Right.
and it's subject to the same complexities and imperfections as any other human activity.
It's important to remember that scientists are people too.
Mm-hmm.
They have their own motivation,
Yeah,
biases,
yeah.
and personal stories. And those human elements inevitably shape the way science is done and the discoveries that are made.
That's why it's so important to have open and honest discussions about the ethical implications of science,
Yeah.
particularly when it comes to powerful technologies like CRISPR. We need to ensure the science is used responsibly and for the benefit of all humankind.
Well said. So we've explored the science, the history, the ethical dilemmas,
Right.
and the incredible potential of CRISPR. But before we wrap things up, I'm going to leave you with one final thought.
Okay.
As we wrap up our deep dive into CRISPR, I'm struck by the sheer scale of what we're talking about.
Yeah.
We're not just tinkering around the edges of biology here.
Right.
We're talking about the ability to fundamentally alter the code of life.
It really is mind-boggling.
Yeah.
And what's even more remarkable is how quickly this field is evolving.
Right.
It wasn't that long ago that CRISPR was just this obscure bacterial defense system that nobody really understood.
Right.
Now it's this incredibly powerful gene editing tool
Yeah.
that has the potential to change everything.
It's a testament to the ingenuity,
Yeah.
the persistence, the sheer brilliance of scientists like Jennifer Doudna, Emmanuel Charpentier, Feng Zhang,
Yeah.
and countless others
Yeah.
who've dedicated their careers to unraveling the secrets of CRISPR and figuring out how to harness its power.
And it's a powerful reminder that science is not static.
Right.
It's a dynamic process of discovery,
Yeah.
constantly pushing the boundaries of knowledge and challenging the way we think about the world.
There's a story in Isaacson's book that really resonated with me.
Okay.
It's about Doudna's advice to young scientists.
Right.
She encourages them to cultivate a sense of wonder,
Yeah.
to stay curious,
Yeah.
to never stop asking questions.
That's such great advice.
Yeah.
Science at its heart is about curiosity, about that desire to understand how things work,
Right.
to peel back the layers of reality and discover the underlying mechanisms that govern the universe.
Right.
And that sense of wonder,
Yeah.
that childlike curiosity is what drives so much scientific progress.
It's about embracing the unknown,
Uh-huh.
about being comfortable with not having all the answers,
Yeah.
and being willing to go on that journey of exploration,
Right.
even if you don't know where it's going to lead.
And it's also a reminder that anyone can be a scientist. Right. You don't need a fancy degree or a lab coat.
Right.
All you need is that spark of curiosity, a willingness to learn, and a passion for discovery.
Well said. So as we wrap up this deep dive into the world of CRISPR, I want to leave our listeners with a final thought.
Okay.
This technology has the potential to reshape our world in profound ways, but it also presents us with some really challenging ethical questions.
Questions about what it means to be human,
Yeah.
about the limits of our power to manipulate nature,
Yeah.
about the kind of future we want to create. These are not easy questions, and
No.
there are no simple answers.
But that's what makes this such a fascinating and important topic.
Right.
It forces us to grapple with some of the biggest questions facing humanity.
Yeah.
Questions about life, death, evolution, and our role in shaping the future.
And ultimately, the decisions about how we use this technology, about
right.
where we draw the line,
Yeah.
those decisions belong to all of us. It's not just up to scientists or policymakers.
Right.
It's a conversation that we need to have as a society.
So, if you're intrigued by what you've heard today,
Yeah.
I encourage you to keep learning, keep asking questions,
Right.
and join the conversation. The future of CRISPR,
Yeah.
and perhaps even the future of humanity,
Right.
depends on it.
We've only scratched the surface here,
Right.
but hopefully this deep dive has given you a glimpse into the incredible potential
Yeah.
and the daunting challenges of this revolutionary technology.
Thanks for joining us on this incredible journey.